Welcome to lecture number 10 of the Cardiovascular section of the Integrative Systems Physiology course. So today we'll be talking about autonomic nervous system regulation of the cardiovascular system. So at the end of our last lecture, what I highlighted were mechanisms that regulated vascular tone, and for several of those mechanisms, there was convergence on changes in the autonomic nervous system and autonomic nervous system outflow. And so when we're looking at sort of a global view of the autonomic nervous system, you can see here, of course, we've got the overall nervous system, we've got the central nervous system, we've got the peripheral nervous system, and critically underneath the peripheral nervous system, what you can see is we've got the autonomic nervous system, which is largely an involuntary system that has input from internal receptors, as we discussed at the end of the last lecture. Many of those inputs are from chemosensitive and mechanosensitive afferents. And then ultimately that provides output to smooth muscles in the cardiovascular system. And that within the autonomic nervous system, that's broken up into two subcomponents, one, of course, being sympathetic nervous motor system, and the other being the parasympathetic motor system. And so when we broadly think about the two functions of these systems, one with the sympathetic nervous system, there we think of that as being fight or flight, so sort of get the heck out of Dodge and redistribute blood flow to where it's absolutely essential. The major neurotransmitter there is noradrenaline, and it's considered to be an adrenergic nervous system, whereas the parasympathetic nervous system is sort of more of a rest and digest system. It increases in gastrointestinal motility and things along those lines, with the primary neurotransmitter here being acetylcholine, and so therefore it is a cholinergic nervous system predominantly. So this is what we will focus on over the course of this lecture, are the details related to these two systems being fight or flight, or rest, digest, and relax. So when we're looking at the autonomic nervous system, again, sort of very high level, you can see that structurally these two are quite different, and of course, in addition to being functionally quite different, so of course the main regulator and integrator here is in the brainstem, which we'll get to in subsequent lectures. But overall, what you can see is that there are with the sympathetic nervous system, we've got a short preganglionic component and a much longer postganglionic segment, and reciprocal structure on the parasympathetic nervous system side. And of course, when we look at broad levels, what are the most important things that we generally tend to think about? Of course, the heart and blood vessels, and to some degree the adrenal medulla being innervated and affected by the sympathetic nervous system. Whereas with parasympathetic nervous system, where we predominantly view that as being integral in terms of the cardiovascular system, is mostly with heart rate and cardiac function. So to put this a little bit more anatomically accurate, you can see here again that what we have is this paravertebral chain of the sympathetic nervous system that houses that synapse point of the pre- and postganglionic neurons, and again with parasympathetic nervous system having relatively long preganglionic neurons and relatively short postganglionic segments. So when we're looking at the transmission of sympathetic and parasympathetic systems in these two sort of segments, if you will, so with the preganglionic component here, sympathetic system comes down. We've got this relatively short preganglionic neuron. We've got acetylcholine being the primary neurotransmitter between at the junction here. And then with the postganglionic fiber here, what you can see is that we now have norepinephrine being the predominant neurotransmitter being released with sympathetic nervous system activation. When we start to evaluate the parasympathetic nervous system, so now we've got this very long preganglionic fiber, we've got a relatively short postganglionic fiber. Acetylcholine now is the major neurotransmitter at both this junction between the synapse between the pre- and postganglionic fiber, as well as at the end organ. So again, a lot of conservation sort of upstream, downstream at the effector organs, much less conservation of mechanism of action. Now, when we look at sympathetic preganglionic neurons, those cell bodies are located in the thoracic and upper lumbar spinal cords from T1 to L2. Those extend to the paravertebral ganglia. Again, as depicted here, so we've got their paravertebral ganglia depicted there, and then those release acetylcholine, which binds with the nicotinic receptors at the ganglionic site. And so when we're looking at how nicotinic receptors work, what we end up having is acetylcholine binding to the nicotinic cholinergic receptor. That results in acetylcholine opening that sodium channel, causing depolarization and subsequent generation of an action potential in the postganglionic fiber. And when we look at ways to modulate this significantly, there's a compound called hexamethonium bromide, which can end up inactivating this channel and can be used experimentally for quite profound impact on the cardiovascular system function. So again, looking at this in a little bit more detail, when we look at hexamethonium bromide, it blocks all sympathetic outflow, and the question would be why, right? And so there, if it's blocking all the sympathetic nervous outflow, what it's doing is it's binding and blocking the activation of those postganglionic fibers at the point of the paravertebral ganglia. So therefore, it can shut everything off downstream of that. And again, essentially what you have is no functional sympathetic nervous system outflow. And during class, what we'll discuss a bit is what you'd expect that physiological outcome to be with a total lack of sympathetic nervous system outflow. With sympathetic postganglionic neurons, now, so those are located again, synapses in the paravertebral ganglia. They are predominantly unmyelinated C fibers, so these are much slower transmitting than what you would have for a myelinated fiber. And those then have axons that extend to effector organs, such as the heart and blood vessels. And as mentioned before, the predominant neurotransmitter being released is norepinephrine. There is some release of epinephrine and other co-transmitters, such as neuropeptide Y, but those are secondary as compared to norepinephrine. So again, when we're looking at in the sympathetic arm of the system, acetylcholine is that predominant preganglionic neurotransmitter, norepinephrine predominant postganglionic neurotransmitter, and we'll just keep on sort of pounding this message home that the conservation at the preganglionic site and at that synapse, and then this difference in the postganglionic and effector organ neurotransmitter as compared to parasympathetic nervous system outflow. Epinephrine can also be released from these adrenergic nerves, and again, these neurotransmitters being ATP, neuropeptide Y, and other things that can result in changes in vascular tone. So when we walk through this a little bit in a bit more detail, when we're looking at the transmission of sympathetic signals, they're initiated in the medulla. They go down the spinal cord to the lateral horn, which is not depicted here, but then they synapse with the sympathetic preganglionic neuron, which here is shown here in red, and those exit through the ventral root to the sympathetic chain. Right, so you can see it's going through here to the sympathetic chain, and then synapse with postganglionic neurons, which are depicted here in dark blue. And so as you can see here, here's that ganglionic chain, and you can see that now what we have is the synapsing at this point of the postganglionic neuron, and then you can see that it comes out through a spinal nerve, goes out to effector organs here. And I think what's critical to note here is that there's not an incredibly fine, refined system here. You can see that this nerve here, it comes in, synapses actually with multiple postganglionic neurons, which ultimately can influence sympathetic outflow to multiple target tissues. And it doesn't have an extremely broad range, but it does have some relative coarseness to how it's going to impact tissues. And of course, when we look at how those tissues are then impacted, of course, it's norepinephrine release we discussed before. It's largely a function of neuronal firing rate. And so what you can see here is under basal conditions, we've got the electrical signals from the neuron here and some tonic level of activity here, norepinephrine being released, maintaining some baseline level of vascular tone. So when you have a change in that signaling rate, for example, an increase in firing due to an increase in sympathetic nervous system outflow manifested as an increase in acetylcholine release at that preganglionic junction or the pre-postganglionic junction, and then subsequently increasing in postganglionic fiber firing rate, what you have is an increase in norepinephrine release and therefore an increase in vascular constriction. The flip side of that is when that threat has passed or when you are sleeping or resting, what you have is a reduction in the firing rate of that neuron, and then ultimately that results in vascular dilation due to reduction in norepinephrine release and binding reduction in the binding of norepinephrine to those alpha-1 adrenergic receptors. So what you can see here now is importantly, while we're looking at this diagram here, we can also have, while we have a relatively coarse amount of outflow to, let's say, multiple regions of the dorsal root ganglia that can impact, we'll say, regions of vascular beds, we can also have feedback from cells and tissues through that dorsal root, which is more of a fine-tuning mechanism. So that's depicted here in these light blue lines here, where now you've got visceral receptors here. So those will be chemosensitive afferents that come back through here, and they end up synapsing all the way back through here onto the sympathetic preganglionic neuron again, as really modulating the outflow of this neuron. So if activity is far too high and vasoconstriction is too great and you're having relative ischemia in those regions, what that will do is that will inhibit sympathetic nervous system outflow to those regions and ultimately enable the vasodilation and the fine-tuning of sympathetic outflow to some of those areas. Now, when we look at in more detail at the example of sympathetic activation at, let's say, the site of a vascular smooth muscle cell or a vascular bed or blood vessel region, you can see that here we've got release of norepinephrine occurring with a frequency or magnitude proportional to neuronal firing rate. Then we have the release of norepinephrine into the synaptic cleft that ends up diffusing to the vascular smooth muscle. We have alpha-1 receptor adrenergic receptor binding here on the vascular smooth muscle. That increases phospholipase C, that increases IP3, that increases increases calcium release from the sarcoplasmic reticulum, and ultimately that increase in cytoplasmic calcium results in an increase in the amount of vascular tone. And so when we look at sort of the mechanisms, alternate mechanisms whereby this is going to occur and their timing, you can see that if, for example, ATP is released as a neurotransmitter or as a co-transmitter, you can see that's going to bind to its receptor over here, to one of these P2X receptors. That results in very rapid depolarization, and you can see here that ATP produces a very rapid degree of constriction here. Norepinephrine, it's a bit more sluggish in terms of its time course for activation, and neuropeptide Y is the slowest to elicit vascular increases in vascular tone here. And again, so what you have is really sort of a variety of mechanisms that enable either quick activation and then some sort of sustained level of vascular tone. So that way you don't have just a simple a singular mechanism increasing a rapid increase in vascular tone. You have multiple mechanisms whereby that can occur and enable better fine tuning. Now, again, when we're looking at sympathetic nervous system outflow and pacemaker activity, that collectively this is a relatively slow response. Okay, so now we're going to shift from the vasculature to the heart. As we discussed previously, what there we have is norepinephrine binding to the, if you recall, the beta-adrenergic receptor. Beta-adrenergic receptor increases in CAMP. That ultimately results in increases in phospholamban activity and increases in calcium-induced calcium release. In addition to that, it does increase the firing rate of the pacemaker. And so you can see here that with stimulation of sympathetic nervous system outflow, what we have is a relatively sluggish increase in the firing rate of the pacemaker. Okay, so here, again, as we'll see later, the parasympathetic nervous system very rapid system. But here, it's far slower and it requires a much longer ramp-up period in relative terms. So now let's shift gears a little bit. Let's look at parasympathetic outflow. So sympathetic, again, being fight or flight. Parasympathetic being the rest and digest system. And so when we look at parasympathetic nervous system, we've got the major parasympathetic nerve for the cardiovascular system we view as being the vagus nerve. So that's cranial nerve 10. And acetylcholine, again, is the major neurotransmitter for both pre- and postganglionic synapses. And so when we're looking at parasympathetic nervous system neurons, they've got long preganglionic fibers. We've got very short postganglionic neurons. The preganglionic neuron has a nicotinic receptor, release of acetylcholine that binds to nicotinic receptors, much like the sympathetic nervous system. And postganglionic is going to be binding to the muscarinic cholinergic receptors. And when we look at muscarinic cholinergic receptors as depicted here, again, they're a 7-transmembrane receptor that is a G protein coupled receptor that when it binds to acetylcholine, the coupling of that G protein to potassium channels ultimately results in the opening of those potassium channels and hyperpolarization of the membrane potential, thereby slowing heart rate. And that is depicted here, where you can see the effects of vagal stimulation on pacemaker potential. As we discussed before, it does really two things. One is it slows the rate of firing, and then two, of course, as we just discussed, we get this tremendous hyperpolarization here that results in a much longer time to reach an action that threshold for firing, and thereby slows the rate of pacemaker firing and slows heart rate. Now, if you recall from just a few slides ago when we were looking at sympathetic nervous system outflow being a relatively slow and sluggish system that results in changes in heart rate. Now, when we look at the effects of vagal stimulation, you can see here preparation with vagal stimulation under normal conditions or unstimulated conditions here, just sort of cruising along. You stimulate the vagus nerve, you actually have an almost immediate cessation of cardiac firing, and that can be a profound repressor of pacemaker potential. And in this particular instance, what you have is the reinitiation of firing here only when you have release of that stimulation. And you can see also not only is there greater hyperpolarization, but then the slope of that increase is also slightly flatter in this recovery phase. But on transient is extremely rapid, and the off transient is also relatively rapid. So when we look at other parts of the parasympathetic nervous system that are important, of course, we've got oculomotor regulation, so that regulates pupil dilation, the facial nerve, which results in saliva production, the glossopharyngeal nerve, which is some airway and some other targets that regulate the vagus nerve, innervating the heart. And so when you look at parasympathetic withdrawal, you say, well, what happens? And you think of times where you're extremely nervous. For example, you have dilated pupils, you've got a dry mouth, you've got an open airway, and you've got a fast heart rate, right? So those are all sort of consistent when you have this orchestrated response of a fight or flight response. That true fight or flight response is sympathetic activation and parasympathetic withdrawal, because they synergize with one another in order to maximize the likelihood of you having a successful escape from a threat. So in summary, when we look at all the things that we sort of discussed today, we've got the central nervous system, here up on top being the somatic nervous system, relatively simple in relative terms, where we got the CNS sending a signal all the way down to the effector organ, whereas the parasympathetic and sympathetic nervous systems, we've got a lot of intermediaries in these, some of which, certainly with parasympathetic nervous system, having long preganglionic fibers, short postganglionic fibers, conservation of acetylcholine at both synapses to modulate end organ function, again binding with muscarinic receptors at the target tissue. With the sympathetic nervous system, We've got these relatively long preganglionic, relatively short preganglionic fibers, relatively long postganglionic fibers, lack of conservation of mechanisms of signaling. So over here we've got acetylcholine being a primary preganglionic signaling molecule, with norepinephrine being the primary signaling molecule at the end organ, binding to both alpha and beta adrenergic receptors, alpha in the vasculature, beta in the heart. And we didn't really discuss it that much, but of course the autonomic nervous system can also increase it can also, through acetylcholine receptor signaling at the adrenal medulla, can increase epinephrine secretion, ultimately being a much more sluggish response to increasing overall vascular tone in the longer term here. But again, just another synergistic mechanism whereby sympathetic nervous system outflow can augment vascular tone in both the relatively short term as well as the long term. But again, the parasympathetic nervous system being extremely rapid, sympathetic nervous system being much slower. So with that, we'll stop there, and we'll look forward to discussing this in class. Thank you.